The central dogma of molecular biology describes how genetic information is stored interpreting the cell. The genetic code of an organism is stored in DNA, which is transcribed into RNA, which is finally translated into protein. Proteins carry out the majority of the cellular function, such as motility, DNA regulation, and replication. Though the central dogma holds true in most situations, there's a number of notable exceptions to the model. For instance, retroviruses are able to generate DNA from RNA via reverse transcription. In addition, some viruses are so primitive that they do not even have DNA, instead only using RNA to protein. DNA contains regions called genes, which encode for proteins to be produced. Other regions of the DNA contain regulatory elements, which partially influence the level of expression of each gene. We think the genetic code of DNA allows both the data about the proteins that need to be encoded and the control circuitry in the form of regulatory motifs. DNA is composed of 4 nucleotides: adenine, cytosine, thymine, and guanine. A and G are purines, which have 2 rings, while C and T are pyrimidines, with 1 ring. A and T are connected by 2 hydrogen bonds, while the C and G are connected by 3 bonds. Therefore, A and T pairing is weaker than the C-G pairing. For this reason, the genetic composition of bacteria that lives in the hot spring is 80% G and C. The 2 strands in the double helix are complementary, meaning that if there is an A on the strand, it will be bonded to a T on the other, and if there's a C on the strand, it will be bonded to a G on the other. The DNA strands also have directionality, which affects the position of the pentose ring where the phosphate backbone connects. This directionality convention comes from the fact that DNA and RNA polymerases in the 5' to 3' direction. With this in mind, we can say that the DNA strands are antiparallel, as the 5' end of the strand is adjacent to the 3' end to the other. As a result, DNA can be read both in 3' to 5' direction and the 5' to 3' direction, and genes and other functional elements can be found in each. By convention, DNA is written from 5' to 3'. The 5' and 3' directions refer to the position on the pentose ring where the phosphate backbone connects. Basic pairing between nucleotides of DNA constitutes its primary and secondary structure. In addition to DNA secondary structure, there are several levels of structure that allow DNA to be tightly compacted and influence gene expression. The tertiary structure describes the twist in the DNA ladder that forms a helical shape. In the quaternary structure, DNA is tightly wound around small proteins called histones. These DNA histones complexes are further wound into tiered structure seen in chromatin. Before DNA can be replicated or transcribed in DNA, the chromatin structure must be locally unpacked. Thus, gene expression may be regulated by modification to the chromatin structure, which make it easier or harder for the DNA to be unpacked. This regulation of gene expression via chromatin modification is an example of epigenetics. The structure of DNA, with its weak hydrogen bonds between the bases in the center, allows the strand to easily be separated for the purpose of DNA replication. This was noticed by Watson and Crick, as it has no escape to notice that the specific pairing that they have postulated immediately suggests a possible copying mechanism for the genetic material. In the replication of DNA, the 2 complementary strands are separated, and each of the strands are used as a template for the construction of the new strand. DNA polymerase attached to each of the strands at the origin of the replication. reading its existing strand from the 3' to 5' directions and placing down complementary bases such that the new strand grows in the 5' to 3' direction. Because the new strand must grow from 5' to 3'. One strand, the leading strand, can be copied continuously, while the other, the lagging strand, grows in pairs which are later glued together by DNA ligase. The end result is two double-stranded pieces of DNA, which is composed of one old strand and one new strand. For this reason, DNA replication is semi-conservative. Many organisms have their DNA broken into several chromosomes, which chromosomes contains two strands of DNA, which are complementary to each other but are written in opposite directions. The DNA before a gene in the 5' region is considered upstream, whereas the DNA after a gene in the 3' region is considered downstream. Transcription is the process by which RNA is produced using a DNA template. The DNA is partially wound to form a bubble, and RNA polymerase is recruited to the transcription start site (TSS) by regulatory protein complexes. RNA polymerases read the DNA from the 3' to 5' direction and placing down complementary bases to form messenger RNA. RNA uses the same nucleotides as DNA, except uracil is used instead of thymine. mRNA in eukaryotic experience translational modification process that edits the mRNA strand further. Most notable process called splicing removes introns, intervening regions which don't code for protein, so that one of the coding regions, the exons, remain. Different regions of the primary transcript may be generated based on different splicing permutations. In addition to splicing, both ends of the mRNA molecule are processed. The 5' end is capped with a modified guanine nucleotides. At the 3' end, roughly 215 adenine residues are added to form a poly-A tail. RNA is produced when DNA is transcribed. It is structurally similar to DNA, with the following major differences. First, the nucleotide uracil is used instead of DNA's thymine. RNA contains ribose instead of deoxyribose. RNA is single-stranded, whereas the DNA is double-stranded. RNA molecules are the intermediary step to code a protein. RNA molecules also have catalytic and regulatory functions. One example of catalytic function is in protein synthesis, where RNA is part of ribosomes. There are many different types of RNA, including transcription initiation, transcription elongation, and transcription termination. mRNA, a messenger RNA, contains the information to make a protein and is translated into a protein sequence. tRNA, transfer RNA, specifies codon to amino acid translation. It contains a 3-base pair anticodon complementary to a codon on the mRNA and carries the amino acids corresponding to its anticodon attached to its 3' end. rRNA, ribosomal RNA, forms the core of the ribosome, the organelle responsible for the translation of mRNA to protein. snRNA, small nuclear RNA, is involved in splicing pre-mRNA, as well as other functions. Other functional kinds of RNA exist and are still being discovered, though proteins are generally thought to carry out essential cellular functions. RNA molecules can have complex 3-dimensional structure and perform diverse functions in the cell. According to the RNA world hypothesis, early life has based entirely on RNA. RNA serves as both as the informational repository, like DNA today, and the functional workhorse, like protein today. In early organisms, protein is thought to have origin afterward via ribosomes, and DNA is thought to have origin last via reverse transcription. Like transcription, in which the nucleotides remain the means of encoding information in both DNA and RNA, when RNA translates into protein, the primary structure of a protein determined by the sequence of amino acids of which it is composed. Since there are 20 amino acids and 1 in 4 nucleotides, 3 nucleotide sequences in mRNA, known as codon, encode for each of the 20 amino acids